Most of us were taught to think of the cell as something like a tiny machine, membranes enclosing molecular parts, all driven largely by chemistry. But every so often, work comes along that suggests the picture may be far stranger and much more interesting than that. Today, I'm speaking with Dr. Michael Hughes, whose research explores a provocative possibility that water may not simply be the background medium of life, but part of the organizing logic behind it. Because once you start looking closely, the cell begins to change shape. Hydration is not just wetness, osmotic pressure is not just a side effect. Phase separation and membraneless structures are not just cellular details. Together, they point toward a cell that is less like a bag of molecules and more like a living physical environment shaped by water, energy, pressure, charge, and emergent order. And what I love about this conversation is that it stays grounded, all while opening deeper questions about what living matter may actually be. Dr. Hughes, it is such a pleasure to have you here. Before we get into your publication, which I will link below, I'd like to start with a little background. Can you share what first drew you into biochemistry and tell us a bit about your educational journey?
Absolutely. Thank you for having me, Dr. Smith. I am really excited to be here and share about uh, what this story I've been building. And so I am spent my time growing up in Colorado in high school. I went to Colorado State University for my bachelor's, and there I had an interest in biochemistry. So I have a degree in biochemistry, and as I was starting this degree, one of my first classes was a cell biology class, and this was really cool to me to start to see the underworkings of the cell, like the mitochondria, the Golgi, all these different parts and organelles of the cell working together. But it was always described as this kind of black box, right? Like one protein goes to one organelle and has some interaction, and then the miracle of life pops out. So I grew to be really absorbed with this black box. What happens when one protein goes and talks to another? And this sparked my interest in a field called structural biology, which is the actual study of the structures of proteins and RNA and these molecules, where their atoms are and what their chemistry does. And so, following this interest, I actually decided to do a PhD in structural biology, and the common ways to find the shapes of molecules at the time was X-ray crystallography. So now there's kind of three main camps: nuclear magnetic resonance, X-ray crystallography, and electron microscopes. And I went to an X-ray crystallography lab where we studied prion diseases or amyloid diseases. And this was really interesting to me because these diseases all have this common feature where these proteins that are these wonderful machines that are doing this black box I talked about in the cell, they have their actual shapes. Like ATP synthase is kind of this hydroelectric dam with this rotating spinning piece that generates energetic molecules. But when these machines break apart and unfold, they expose parts of themselves that can aggregate and stick to each other. And in some instances, you can form a fiber, and this protein fiber can be really hard, like even the stiffness of a copper wire. So you can imagine that your body can have a hard time getting rid of these. And this phenomenon of forming these long protein fibers that we call amyloid fibers is associated with Parkinson's disease, ALS disease, Alzheimer's, all these neurodegenerative diseases, mad cow prion diseases. They all use this special kind of protein fold when things fall apart. Um, and Colorado State University actually had a chronic wasting disease separation or IRIVIS research. So I actually had background in my undergrad into this idea. So I was able to hit the ground running with these ideas when I started my PhD. And this is kind of what started to get my whole journey going, was that when I learned about amyloid, it was always in the state of something wrong, a protein unfolding, going awry, something bad happening in the body. But when I started my PhD, I came across this odd area where people were starting to discover how amyloids were useful. So one example is bacterial biofilms harness the amyloid fold to make their kind of biofilm home that makes it really hard to get rid of infections because they have this really, really nice space that they build. Or spider silk is another version of this. So the amyloid fold actually then, at the start of my PhD, kind of got to blossom into this idea of something useful as an organization trait. And that's actually how my PhD project started, that kind of unfolded to the work I do today. But overall, my interest has always been trying to figure out the structure of how atoms actually move and fit together to carry out biochemistry in the cell.
So, was it your research work that ultimately got you interested in deeper questions regarding the cell and perhaps water?
Absolutely. So I started to become an expert in protein-protein interactions, which kind of dictates how proteins will talk to each other in this black box I alluded to earlier and transfer information or do enzymatic reactions and all the things that we need to carry out. But this lens still seemed incomplete, didn't quite explain some gaps or just big ideas, right? It's hard to go from these two proteins interacting to maybe a big sweeping genetic disease view of the body, where one gene missing can just, move a little piece from thousands of machines around the body, and it can be really hard to figure out exactly what's going wrong there. And so there's always this thing I was missing as I was trying to go through and understand all these biological systems. But the nice thing about the structural biology view is that's very nuts and bolts. It's very hands-on. Things aren't mysterious. There's actual movement there. So I began to realize as an expert in protein-protein interactions, I couldn't explain everything I was seeing by just looking at how the proteins talk to each other. But whenever we're doing experiments or these proteins are in the cell, they're surrounded by water. I realized that's the one thing our experiment is always removing, and all of our pictures are always removing, is what the water is doing. And so I kind of got this hunch that that spot I was missing for understanding how all these interactions take place was missing this water component that I wasn't seeing. So throughout my PhD and towards my postdoc, I began to become really interested in understanding what water is doing in biological systems and how maybe missing that could help fill in some questions I had about biology and biochemistry.
Yeah, your paper really highlights some of the more anomalous or unusual physical properties of water. Could you describe what makes water so special in biology and why it sometimes behaves, I guess, in ways that don't seem intuitive?
Absolutely. So my first attention to this is I had this lucky break. I almost forget how it happened, but I came across this website by Dr. Martin Chaplin from London Southbridge University, and it's kind of this underground water mecca where he's accumulated and listed these 60 to 70 water anomalies. And these are anomalous properties of water where when you compare water's behavior in this situation, it is really deviant from most other substances. And that's kind of like this really generalist, nebulous term, but most other substances might be like organic solvent. Does olive oil do this? Does butter do this? Does cyclohexane do this? The, all these are kind of comparing to lots of other liquids that we don't come across a lot, but water is just this one liquid that's ubiquitous everywhere in our lives that has these strange anomalies. And these anomalies happen to have these things where water is kind of backwards in its behavior when these other substances are having this normal behavior. So the one I like to point to is when you cool water, it expands, and this is really rare. So if I cool an oil, it'll always shrink, and then at a certain point, it'll kind of freeze like an ice cube, but that will be smaller. So a water, as you cool it, it has a density maximum around 4 degrees Celsius, but every degree you cool from 3 to 2 to 1 to 0, you actually expand your volume after on that property. And so this would be an example of an anomalous property where water behaves differently from most other liquids that we have. And this expansion of volume is really interesting because, first of all, when we're talking about physical work, it's always pressure volume work, right? If you have a steam turbine and you're doing industrial revolution, we're doing pressure volume work in all of our engines all the time. So there's an immediate kind of thread there to pick up on. But then there's another one where water begins to get more ordered because that expansion of volume comes from this tetrahedral. Water starts to kind of click and lock into this tetrahedral shape where it optimizes its 4 hydrogen bonds. Whereas normally in water, there's 3.7 per water molecule. If every water molecule can accept 2 hydrogen bonds and donate. But as you cool and you start to get all 4 more commonly satisfied. And this kind of pushes out some. So water has this strange property where it can kind of turn its volume into order. And this has immense consequences for things like entropy and information as we have our modern view of science, right? And are starting to appreciate what these words might mean. So there's this weird tie of water's entropy to temperature that's backwards. from a lot of other substances in the temperature regime that life happens to hang out in. From around 0 degrees to around 60 Celsius, you'll find a lot of these anomalies occurring.
Does this explain some of the kind of strange things we see when water meets a surface, like a membrane, a macromolecule? Because it seems to behave in a different way than bulk water. So, what has your research shown in this context?
Something strange does happen when water touches an interface or a surface. And we tend to call this the hydration shell when we're talking about molecules or small things. But most intuitively evident is the meniscus that forms when you look at water in a glass, right? Water kind of like slopes up to the surface. So immediately, without any prior knowledge, and if we have very pure water, we have this strange mystery where it starts to like build itself up and structure itself up the side of the glass. And if you imagine like that meniscus is small to us, millimeters, but is hundreds of thousands of layers of water molecules at that level. So there's some structure seemingly communicated from the glass deep into the water. And this in this particular case, you think we seem to get more tetrahedral ordering, right? So water has this response to when anything touches it. In this case of the glass, but then when we put anything, dissolve anything, and that might be sugar or protein or salts into water, it will have this consequence where the water shells around it are immediately disturbed compared to what they would normally be, kind of like when the water is touching the surface of the glass. And these are different for each kind of molecule and multivariate. And I realized that this hydration shell was that piece I was missing. There's a strange behavior where when you're looking at the water behavior in the middle of the glass, it's different from the water behavior at the side in the meniscus. And so by extension, water in the middle of your test tube is different from the water immediately on the surface of the protein in your test tube. And this is the origin and the interest of the hydration shell that I talk a lot about in my paper.
Yeah, so the hydration shell is an important aspect of biology, essentially.
That's right. And it has always been known about. People have and scientists have talked about this for a long time. In the case of the glass, it's hundreds of thousands of layers of water that are disturbed to make this meniscus. But in the case of a protein, oftentimes we only measure one or two layers. And the kind of strange thing is that scientists have known about this hydration shell, but as our tools evolve, our ability to interrogate this hydration shell has changed. And I think that my paper is a very contemporary look at what has been added in the past 20 years about what these hydration shells look like and trying to tie it back to biological systems.
Now, you present osmotic pressure in a way that feels much more central than it's usually taught. How would you describe the basic concept behind osmotic pressure, and why is it so important to the ideas we're about to explore?
I think that osmotic pressure is one of those, like if I had a tagline, I'd like to say like osmotic pressure is the dark matter of the biological universe, right? For appeal. But it's kind of this thing that we know about. We've always learned about it, or commonly in high school classes. But then I entered into this realm of biology and biochemistry at getting my PhD. And I didn't talk a lot about it in the sense of in the cell or in a living system, that there's all these particles, there's an osmotic pressure that's exerted. So when we're talking about why does a cell like to move somewhere? Well, we as a biochemist or a cell biologist might talk about activating motor proteins and signaling to identify what's attracting or what's pushing it away. And all of that is absolutely there and a really valid lens to use. But another one is that to actually like move, osmotic pressure moves things as well. And there's always these changes and needs for balancing of osmotic pressure. When we imagine the U-pipe experiment where you kind of have water in a plastic U-pipe and there's a semi-permeable membrane, which the definition of a semi-permeable membrane here is it lets water across, but it doesn't let what you've dissolved in the water, whether it's a protein or salt or sugar. And so you can pour like sugar in one side of your U-pipe and then that side will raise. Because the dissolving of the sugar into that side exerts an osmotic pressure, which then happens to draw up water from the other side to balance. And so you have like an elevated water level. And so when you get that new equilibrium, the osmotic pressure is the amount of, like gravity pushing down on that elevated level. And so this is all like in the context of a U-pipe. But in the context of a cell, we don't really learn what that does. And so why I talk about that is because the cell's nuclear envelope is a semi-permeable membrane, right? It won't let the DNA out. It won't let some things from the cytoplasm in. How does an osmotic change that? Because these molecules their movements are dictated by the rules that osmotic pressure helps set out. Right? It's not always just a signaling pathway or a motor protein. It's these physical phenomena like osmotic pressure that dictate where molecules move in these systems as well. So even immediately then looking at like gene regulation, osmotic pressure, and this concept of the nuclear envelope being a semi-permeable membrane now should be influence how we think about genetic expression and these kinds of things as well. So that's how I start to tie in why we should be thinking about osmotic pressure more seriously when interpreting the behavior of living systems and understanding them.
Definitely. It reminds me of one of the experiments that you discussed in your paper. It involved a cell membrane being disrupted, and yet the cell does not simply just lose its organization the way we would expect. Could you walk us through the findings and what they suggest about how the cell is actually organized?
Absolutely. So this phenomena you describe really blew my mind when I came across it, like in my 30s, as a professional practicing scientist already. It's like that can happen. So the idea is, if you have just a bag of enzymes and I cut that bag open, these enzymes just spill out and dilute that into the system. If we think about a cell like that, when you cut the membrane, which holds this bag of enzymes together, we expect it kind of just spill out. But that's not what happens for some cases. It seems that there's certain cells where you can induce a pore in the in the cell membrane, and it actually not only maintains its shape for hours, if not days, even hours, that while it's maintaining its shape, it's still actually looking like a living being. Scientists have been able to show that the cell is still like functioning, and they have even shown that they can get rid of the cell's membranes. They basically can take cells, put them in a blender, blend them up, get rid of the membranes, but have nuclear, the nucleus present, and they will make like pseudo or proto cells where you actually look at something that looks organized like a cell around the nucleus, and these can undergo division, and all with a super disrupted membrane. So
That's wild.
right. Where do we put that? Right. This sounds absurd given what most people have learned through whatever classes and what I learned. So these papers have been published here or there, but I don't think that we focus on what that means or how to interpret that. And so I really like describing this as a point to why would someone care about the paper I wrote? Well, I try to approach and explain why that phenomenon can occur in a reasonable way and in a way that might help us understanding. But that's my approach to it. But there's something that I left out of my paper that I actually like that you asked I can address, is that this phenomenon isn't the case for all cells. Some cells, when you pop them or puncture them, they do pop or dissolve like a bubble. And I wish in my paper I'd highlighted that both of these exist, because I think that like a fruitful area of research would be to what cells do maintain their structure and shape, whereas what cells do really rapidly fall apart. And that's something I didn't get to in my paper that I wish that I could go back and include. But just the idea of taking this into consideration of cell organization really, really demands that we try and understand what's going on behind this phenomenon that seems so strange and surprising to us.
Definitely. I can't wait to hear more about that in the future. For people who are new to liquid-liquid phase separation, what is it in the most intuitive sense, and why has it become such an important idea in cellular biology?
Right. So where this story about liquid-liquid phase separation will end is back at the cells staying together that we just learned about. But where it started for me was during the beginning of my PhD. I had this very strange experience because I went to the structure lab to do X-ray crystallography on amyloid proteins and figure out what the amyloid fibers look like, so then we can start to design molecules and drugs to counteract that, to dissolve them. And the major idea is that when we get a picture of what this looks like, we can just much better understand how to resolve the complications that show up in the body from this. And so that was the promise. But then, as I also alluded to, I kind of came at this time where we're starting to appreciate that all of this can be functional and not just detrimental. So having these ideas in my head, I started on this paper which showed that this normally ALS-related amyloid protein fuss could form amyloid fibers, but that they were really easily reversed by heating a little bit or by like SDS. And normally these protein fibers involved in amyloid diseases are ultra stable. And you have to like boil them or dissolve them in pure sodium hydroxide, something like that. So this was a really, really labile, as we call it, like reversible fiber, and that was interesting to me. So as the field developed, it was found that FUS could undergo this thing called liquid-liquid phase separation, where it would, if you took your solution of protein with FUS in it, or HNRMPA1 is another really famous one, and you cooled it, it would form these droplets in your solution. And people always compare this to like oil and vinegar and salad dressing, right? It's two liquids, but they, if you shake it up, you can see them phase separate as the oil blebs out from the vinegar. So that's kind of like what's happening here. The trick is that there's water on both sides. It's liquid phase separation, where as you cool your FUS solution, the protein FUS forms a dense liquid droplet. Where there's more FUS in the droplet than outside. And so what's so strange is that, like, even as you're removing energy from the system, you're creating this spontaneous organization of molecules, right? And there is like a callback or a tie-in to what I was talking about earlier with the weirdness of water. Is as you cool it, there's this strange like spontaneous organization into tetrahedral hydrogen bonding. And so, this concept comes up, but it was very strange as biochemists began to see this, especially with these amyloid-related proteins. They were then found in the 2010s to 2020s that many of these ALS-related amyloid proteins will undergo this phenomenon, that before they become an amyloid fiber, they undergo this liquid-liquid phase transition where they form this dense substrate, and then amyloid fibers show up later down the road. So this was really captivating to biochemists and kind of is changing the field right now as we speak. I don't quite know how it's like spilled out of the ivory tower and in which ways, but this will start to seep into us, because what was found is that these proteins will use this liquid phase separation principle in the cell to organize as well. And this is actually famously shown by a Professor Cliff Brangevine in Princeton, who's at Princeton now, but he had a paper that showed in C. elegans, which is a worm, as the embryo develops, there would be these droplets that would form, these puncta, these liquid drops would form as the symmetry of the embryo is broken, as you have a head and tail of the worm that has to be dictated at the molecular level from the embryo as it builds out. So what was so strange is that on one half of this worm, there would be this spontaneous organization of these droplets, and then on the other half of the embryo, they'd be absent. But what is so strange about this phenomenon of liquid phase separation is you're not wrapping these molecules in a membrane. So when we think of the mitochondria, the nuclear envelope, the cell itself, it's always wrapped in a membrane that creates this partition that will keep what's inside. And here there is no membrane. There's just two different liquid environments. One has a different assortment of molecules than the other, and this is spontaneous, just physically emerges from the system. So how do we explain that? What do we make of that? And the next thing we know is that the cell is using this actively for regulation and control of how it's going through life. And then where this ends is then we can kind of think about, why does the cell itself not fall apart when I disrupt the membrane? And now with this idea of liquid phase separation, where you can have these molecules spontaneously organized into a cohesive condensate or liquid phase, that when you get rid of the membrane, you don't need it to fall apart and spill out anymore, right? It's just a whole new paradigm of organizing molecules in the body. And that's what's so powerful about this lens.
Yeah, I guess I never gave much thought to what I was actually understanding membraneless organelles to be, because I just somehow pictured that there was still something like a membrane around it. But it sounds like what's really going on is this liquid-liquid phase separation, allowing some sort of order to emerge. Is that correct?
That's absolutely correct, how I see it. So in the common way that the body will use this is one of the biggest, most famous ones. So before this field kind of blew up, the word membraneless organelle was often referred to the part of the cell called a ribosome. So a ribosome is a huge machine that turns that makes your proteins. And so if there's DNA to RNA to protein to ribosome, this sits between RNA and protein, turning the RNA messages into protein. And it's not wrapped in a membrane. It's just a huge machine floating out there. So we call that membraneless organelle. But then after this kind of revolution and acknowledgement of this liquid phase separation phenomenon, we start to see that all of these like puncta in the cell, that whenever you're looking with a microscope, you can start to see these puncta organizing, and they seem to obey this liquid-liquid phase separation principle. And what that means is that there are some principles in physics that dictate how you get liquid-liquid phase separation. And this is actually like figured out in the 50s and 60s and 70s by polymer chemists. So chemists always knew about this. And like if you're trying to emulsify or package shampoo, there's some processes of like drug creation where you might want to have a liquid like separation to capture your drug or have that for delivery. But the weirdness was applying it to the cell. And so a famous one before is the nucleolus. The nucleolus is the area of DNA inside the nucleus that makes the ribosomes. It's where the DNA that turns into a ribosome lives. And this this is a very unique environment. It has a different pH than the surrounding nucleoplasm. It has different molecules on average. It has like different DNA on average in it. So it has all these unique parts about it that sequester for unique identities of molecules and makes this unique environment that's not wrapped in the membrane. And so again, it's hard for us to understand. So what this is pointing to is things can enter and leave the nucleolus quite freely, right? It's just, can you disentangle yourself from the molecules that are holding you back, all right, in the nucleus or in the cytoplasm and go across, whereas the membrane is more like a barrier. So what the membrane is shown to reveal to us is it's just a barrier that takes longer for things to move between on their way to balance things like osmotic pressure or electric potentials that dictates where molecules go. So the membrane is something that just slows that down. And then you can even get fancy like life does and add pores that like specific things cross at certain times, but not others. And so you then can gate how permeable you are to other things. But the liquid phase separation, you're in a sense always permeable, right? It's just biophysical equilibrium that tells you if things are getting pushed into you or pulled out of you. And so changes in biochemical equilibrium then can be realized as like temperature changes, pressure changes, all these physical things. And so we can start to incorporate that into our thinking as well about how that is. But it really points to a membrane as this additional gate layer between two different areas of the cell.
So it seems like depending on what organelle you are, what your purpose is, sometimes you would want to be more permeable than other times, from what I'm hearing. And what I'm curious about, that makes a lot of sense. Thanks for explaining that. But what I'm wondering is what actually, I guess, maybe at a level down, is driving this emergent state? What allows it to not just form, but then to, well, form, but then to stay structured in a sense?
Yeah, this is a great question. So the field of biochemistry has really like dove into this in the past 10 years since this kind of showed up in the early 2010s and to the 2020s. So the idea that emerged is that when we use what's called multivalent interactions, and this is basically saying that a protein is a polymer, meaning that it's many things tied together in a long string. And multivalent means that there's many ways that they can interact with each other. So like a hand is just like single valent, but then as soon as you have like another hand, you can make like another interaction. This would be like a dimer, a protein-protein dimer. But if you had a string of my hands together, and now you can imagine how I could like make a tangled mess of my two arms, like interacting through all these different hands. So that becomes this multivalency, and you can see how it immediately leads to this like knot or kind of many long RNAs and proteins interacting with each other. So the emergent property that was thought to drive this is basically like small hydrophobic interactions or like opposite salt pairings that we call ionic interactions, like a positive salt on the protein interacting with the negative salt backbone of RNA could be one of these multivalent interactions. So we start to piece it together in this standard frame I had as a structural biologist of looking at the protein structure interactions that hold it together. So you can kind of see that maybe as you like cool the temperature of this example I used earlier of a FUS protein solution, that at a certain point when it's cool, you get these multivalent interactions or these like hydrophobic interactions, and the energy of the molecules jostling around in temperature can no longer break that interaction. So as you cool, you can't overcome this slight attraction you have to your neighbor molecules, and you start to form this tangled mess, and that's the protein condensate. So it's just like a lot of little handshakes between molecules that becomes energetically favorable under some change of conditions. It's just the abstract way to say that.
So, what another way of saying that is that it's a thermodynamically favorable state?
That's absolutely correct. Right. So uh, this arises just by a solution equilibrating towards this thermodynamically most stable energetic state. Right. The balance between like the energy and the entropy of the system pushes the molecules together in this strange way. And the biochemists, would tend to describe this as by the interactions between the proteins.
You sent me a really interesting publication about stress granules, and they seem like a beautiful example of the cell temporarily reorganizing itself under stress. For people who are unfamiliar, what are stress granules? What role do they play in protecting or regulating the cell? And what happens when that normally reversible process becomes persistent?
Absolutely. So the stress granule is one of the most famous and commonly studied membraneless organelles that has emerged out of this field of research. And what it is uh, if I have a cell that I stress, and stress is just a really general term for a change of conditions, right? Like if I move to a new city, I might be stressed as a human for a little bit, right? But at the cell level, this might be like a heat shock. So changing the temperature from 37 degrees to 43, kind of like if you had a fever, or but way faster, right? It's not exactly like that. And then another might be like a slight poisoning with sodium arsenate's one that we use, or maybe energy deprivation or osmotic stress of having a really sugary solution poured on you suddenly. And so all of these different stresses to the cell, some of the proteins in it will have the same behavior, where you will find these spontaneous formations of punctas or dots in the cytoplasm of the cell, where stress granules form in response to this. And stress granules are we don't really, I would argue, we're still trying to find out what they mean biologically. So can't say what a stress granule like means for the cell, but what they have from our observations is they're enriched for our mRNA, certain mRNA. So mRNA is the message that the nucleus sends out of RNA from the DNA that encodes proteins that the ribosome will turn into proteins. So immediately when you get stress granules, you get enrichment of mRNA and some mRNA binding proteins that hang out. But these used to be kind of diffusely floating through the cytoplasm. So what's strange is that they spontaneously organize with each other under this stressful change of conditions. And they form this membraneless organelle that, as the stress passes or the cell gets used to it, it starts to dissolve, and they release all the mRNA and the proteins back into the cytoplasm. And what the explanation that the biochemist might come up for to describe how this is useful is that when you encounter a stressful situation, you need to change your behavior, right? So if I move to a new city, I need to pay attention to make sure I keep it on a schedule, developing a schedule, finding friends, food, water, everything that might keep me alive, right? So it requires a change in behavior. So when the stress granule forms in the cell and you get all these mRNAs concentrated there, they aren't free to be translated into proteins like they might normally have been, right? So normally when we're talking about changes of DNA expression of like what proteins the cell is creating from its DNA, this is a question that really matters because it tells you what kind of cell you are. Are you a cancer cell? Are you a normal cell? Are you a neuron? It's all what proteins you're making. So, but this can take out days and weeks to change that of a cell as we grow. But a stress granule, it forms on the order of minutes, right? So you can now change the expression of what the cell is doing with its genetic information just by inducing the stress granule and sequestering those pieces away. And this can be great because then the cell can now use that energy that it's saving from making this program and put that into maybe a stress response or recovery program. So that's how we might argue that these are useful, right? But at the end of the day, we know that stress granules are little inhomogeneous spots where certain proteins like to be under change of conditions and release later. And this got tied into amyloid research because a lot of these stress granule proteins are these proteins like FUS, these ALS-related proteins that also form amyloid. So for a long time, the thought was that the stressful conditions, if they persist, might gather all these proteins together in this concentrated soup, and then that might lead to, if it's persistent, the formation of these amyloid fibers that lead to these diseases, having them all interacting with each other. And frankly, this has kind of like fallen apart over the years as we figured out more and tried to like prove that. It's more complicated and stranger, but these were the ideas that the field had developed around the stress granule, what it is, what it means to the cell, and what it represents.
One thing that stood out to me in your work, among many things, is the fact that we tend to give separate names to things like pH, ion gradients, membrane potential, even osmotic pressure. But it's starting to look like beneath it all, they may have a deeper electrochemical layer kind of running through all of them. What are your thoughts on that?
Yeah, so this question is where I start to try and tie a lot of ideas that we've always like known about, but together, tie them together in a way that we can start to understand the cell in a more biophysical way. And what you talk about with, we have these things for pH gradients, ions, membrane potential. So these are tied to the concept of electricity or bioelectricity. And frankly, upon coming into this field and having these concepts thrown about, I wasn't an expert on them. I actually had a joke from a professor in college that no professors actually get to the electrochemistry chapter because they don't know it. They don't have to teach it. And so here I was, right? The same one, not knowing. And why it came up to be is because when I mentioned this membraneless organelle, the nucleolus, earlier, I mentioned that it has a lower pH than the surrounding nucleoplasm. So the nucleus is slightly acidic, usually, maybe like 6.8. And then the nucleolus at its center is like pH 6.5. And this so earlier I talked about how osmotic pressure comes from water trying to equilibrate this pressure. And you can actually like have water be sucked up this one half the U tube, this U pipe. And so it represents a strong physical force. So inside the nucleus or the nucleolus with the low pH and outside, we expect actually that the osmotic pressure is being balanced. That as soon as some solute leaves the nucleolus and that osmotic pressure decreases, something should be moving back in to fill that void. So the osmotic pressure will constantly be balanced. But why is it that if that's the case, that the pH isn't balanced, right? Because pH comes from the ions that we dissolve into the water. So if I pour hydrochloric acid into water, I'm adding the hydrogen ion, I'm adding the chloride ion. And it's increasing the osmotic pressure because I'm adding dissolving stuff in it, but it's also changing the pH. Now, if I add potassium chloride, I'm adding two ions, so I'm increasing the osmotic pressure, but it doesn't change the pH. So what's strange about the membraneless organelle is that it is balancing osmotic pressure, but at the cost of making a permanent pH gradient, right? So now we're turning like balancing an osmotic gradient into generating an electrochemical potential, a difference between the inside of the membraneless organelle and the outside. And so suddenly electrochemistry became central to understanding this phenomenon and really trying to understand what's dictating how molecules move. And when we're talking about how molecules move, we're really talking about how ions are. Our proteins and our RNA, they have all these charged groups on them. They're ions, they're polymer ions. And there will be these rules where certain like proteins that are in the nucleolus will have certain ions that they prefer to have on them than outside. So now through this lens of what molecules are enriched in outside and inside, we get to see how they are establishing and maintaining this pH gradient. So the pH is a function of what's dissolved in it, of what is dissolved in that water at that area in time. And so what this opens up is how the concepts of pH, ions, membrane potential, and osmotic pressure all talk to each other through what I would call electroosmotic, right? The idea they're trying to reach electroosmotic equilibrium. So the thermodynamics that the cell is trying to get to is to reach electroosmotic equilibrium, where the pH gradient or the ion gradient and the osmotic pressure gradient are all balanced and at the lowest energy level. And the goal of a living organism is never to ever get there, because if you get to that, you die, right? Suddenly life starts happening. So there's something weird about the living organism is always keeping you away from equilibrium, but this phase separation process is by the system trying to return to equilibrium.
I'm assuming that in order to keep that state out of equilibrium, we're consistently requiring the use of energy. Is that correct?
So as we know life, right, we want to ingest energy and we use that energy somehow to keep us in electro-osmotic disequilibrium. Uh, and our bodies, all of our processes that we're observing are on some way, like as we understand it, the molecule is trying to get back to electroosmotic equilibrium. But in the food that we eat, we can see that there's energy stored there. And there's also this strange way of looking at metabolism that I feel is underappreciated given this lens is that when you turn one glucose molecule into six carbon dioxide molecules, how is the osmotic pressure of your system changing through metabolism, right? You're taking one glucose and six oxygens, and then you're turning that into six carbon dioxides and six water molecules. And immediately you're playing with particle number, which is osmotic pressure. And so kind of like wondering then how is your body this weird like osmotic pressure generator equilibrator, and how are you using that for harvesting energy and harvesting energy gradients? So most generally, systems harvest between energy gradients, right? The food that I enter in and whatever I excrete out, there's an energy gradient that's driving everything inside.
That makes me think, talking about energy, about the mitochondrial example that you had in your paper. And I think it was that the calculated number of free protons, that they seemed far too small, as in not enough, compared to what was anticipated to be needed, if I'm understanding it correctly. What do you think that paradox is telling us about protons in biology?
Absolutely. So this is a really interesting little bit that I came across by a small paper that was written about how unintuitive pH is when we most think of it. So uh, we just addressed how pH and ion gradients are tied. What is creating your pH gradient happens to be the ions that you have dissolved in it. So in the case of hydrochloric acid, this makes a lot of sense because we're just adding hydrogen and chloride, and this hydrogen goes off and enters into the water, the aqueous phase, as what we call hydronium, because it kind of burrows into a water molecule and turns it from H2O into H3O plus. And this is how acid exists in water, and as an ion in solution. So uh, what pH is very technically the log of the concentration of hydrogen. So it's a way to represent the concentration of the number of H plus dissolved in your aqueous solution, the number of that ion. Uh, so that's what it's taught to us as. But how do we measure pH? We would use a pH meter. So what is the pH meter measuring? It is not actually the number of hydrogen ions. It is actually the millivolt potential, the difference between your reference electrode, which is usually a salt solution that's like kind of like 3.5 molar potassium or silver chloride, uh, and your measuring solution. So the difference between your reference and what's there. And so there's a millivolt potential difference between these two. And this is kind of similar to how when I'm talking about there's like in the nucleolus, there's a pH different than outside. But what is counterintuitive is that then so what the pH meter is measuring is a millivolt reading of some electronic that we have. And then what we do is we take that and we convert that to equivalent units of the hydrogen ion, and that becomes pH. So pH becomes how many numbers of hydrogens would you have to have dissolved in your solution to reach this millivolt potential that the pH meter measures. So what's strange here is, first of all, we're measuring millivolt, and then we're mathematically converting to hydrogen. The issue with these conversions that we do, whether it's calculating osmotic pressure or the pH level, is that we're assuming what we call ideal solution conditions. And this is kind of, in physics, there's a joke about like a spherical cow. That's the ideal thing, right? If you're modeling something, you turn it into a nice, well-behaved sphere, and it has, not a whole bunch of weird friction arguments and trying to dictate its motion. And we do that in biochemistry when we're calculating osmotic pressure. We call it an ideal solution is when the particles don't interact with each other in a way that can mess up your measurement. Because osmotic pressure stems from the number of dissolved particles, but what if the two particles like interact and become one, right? Now you have less osmotic pressure. And in the cell, this kind of looks like that tangled mess that I was talking about, all these multivalent interactions, like when an RNA binding protein interacts with RNA, and you form that stable interaction, you're really like decreasing the number of particles, right, by kind of becoming like one bigger one. So this is what non-ideal systems do, which is all the ones we look at, all of life, all the things we're interested in, they are not ideal. So when we're looking at pH measurement, it's consuming an ideal thing. So when these authors are looking at the volume of the mitochondria and they calculate how many, at the reading of like pH 5, how many free protons do you expect to exist in the nanometers of volume enclosed in the mitochondria? And the answer they come up with is like 7. And so somehow these 7 numerical hydrogens are driving like thousands of generators of ATP synthase on at the same time, right? So it's not exactly the particle we're looking at, right? What we're really measuring is a millivolt potential difference between solutions and then converting. And so if we don't keep that in mind, then we end up in these weird situations where we try to convert to like number of hydrogens to understand the particles, and the particles makes absolutely no sense given the other number of particles that they're powering or driving. And this is a consequence of because of how we view and set up our experimental systems and assume ideal conditions even under times when they're not. And the cell is not an ideal solution. It is not fulfilling those conditions. And so this would force us to look somewhere else. And the what I pose instead that we're looking at is that pH is a measurement of the distortion of the hydrogen bond network that as a consequence of the ions dissolved in it. And that's kind of what I reframe as we could pay attention to, as opposed to just strictly the pH number. But however it is explained, whether it's mine or another explanation, is still left with these strange conundrums highlighted by this example, the mitochondria.
Yeah, everything is so dynamic and seems to have way more roles than I originally even imagined. And that brings me actually to ATP, which was my next question, because we usually just think of it as being the energy currency of the cell. But you suggest that there might be some other things that ATP is doing.
That's right. So ATP is often taught as the energy currency of the cell, and we use this in that the mitochondria makes ATP from our metabolism, digesting the glucose that I talk about, and then our other systems burn ATP, whether it might be an enzyme trying to do a chemical reaction, or most commonly, like my muscles waving my arm around. That's all ATP motors being driven. So ATP is usually thought of as this energetic bond that the ATP stands for adenosine triphosphate, and so adenosine monophosphate is actually what RNA is built of. It's just a bunch of those. But there's something about turning diphosphate into triphosphate, ADP into ATP, where you put on this third phosphate, and it's just a really energetic bond. ATP is not like a stable molecule, so we think of harvesting that unstable bond as energy for the cell when it breaks apart. But it turns out ATP does a lot more than this. If you start to deplete ATP in the cell, the cytoplasm turns from something liquid into something glassy in bacteria. Suddenly, rearrangement of molecules slows down and stops happening. What is going on with that? Well, it turns out ATP has the same interactions as soap or detergent does with water and how it changes its hydration shell. And so ATP actually is kind of like detergent, where it keeps your body like liquid in a weird sense at high concentrations. So some strange ATP facts, which I'm now a huge connoisseur of and love, is that the motor proteins that drive your body, they interact with ATP very strongly. And so the measurement that we use as biochemists would be calling this micromolar, right? If you have like micromoles, which it's like, I think that'd be a million, a millionth of a mole or something like that, right? That ATP will stably interact with your motor protein binding at that concentration. And so when we deplete the cell's ATP is normally in millimolar, which is like 1 in 1,000 or something. So it's actually one of the most abundant molecules in your body. So the most numerous molecules in your body by number is water, potassium ion, and then at 200 millimolar, and then ATP at like 3 to 4 millimolar. So your cells will actually keep ATP concentrations from 7 millimolar to like 2 millimolar, but below 2 millimolar, you'll start to get stress granules and the bacteria with a glassy cytoplasm. So things stop to rearrange and stop moving around each other in a fluid motion when you decrease ATP. So it has a strange interaction, kind of like dish soap facilitating this. But there's an even stranger bit that I can tie into earlier when I talked about poking holes in the cell and having it stay whole. I mentioned this experiment where they can actually like grind up cells in a blender, and as long as they put the cytoplasm and the nuclei back together, they'll get these even self-dividing uh, cell-like blobs. It turns out this only works if you have ATP and also a motor protein available to work. But you have to have 4 millimolar ATP or a high concentration of ATP. If you don't have that, you don't get the spontaneous organization of the cell. It doesn't work to reorganize. And so there is something by mere presence of ATP, you happen to have the cell be able to self-organize in a way that we know and appreciate, where it's always fluid and moving around itself. And then when you deplete ATP, things go awry.
Interesting. Now, when I think about ATP, just because of all its phosphates and how negative it is, it surprises me, honestly, that it has this organizing effect. Is it because of the kind of abundance of negative charge on it?
So I think that this is an open area, and I like how your brain is approaching this by looking at these biophysical aspects of it, because that's what I noticed as well, is that it's strongly negatively charged, and there's also things that are weakly negatively charged. But a lot of the ions, and my paper talks about this and how we categorize them, but there's this strange, I would say like what do I call this? Like a dictionary. There's a strange language that's encoded in the molecules of your cell that dictate how they interact with each other. And so, strong negative charges like ATP or RNA is, and also all of your proteins are covered in strong negative charges. They interact with water in a particular way. It's the opposite of positive charges. But the thing about positive charges is that if you have a strong positive charge, you can interact directly with the strong negative charge and push out all the water. But if you have a weak positive charge come in and interact, the weak positive charge actually can't move. It's not energetically favorable to move the water away from the strong negative charge, because the water is so strongly attracted to the negative charge on that phosphate in the ATP example or the RNA example, that a weak positive charge can't get past that. So a weak positive charge is going to be, in the context of the cell, a potassium ion. That's one of those most numerous ones I talked about. But a strong positive charge would be calcium or sodium. The ions that are released when action potentials like a muscle contraction or a neuron is firing can strongly bind, and they can push water directly out of the way and bind directly to the phosphate. And so ATP is really interesting because it has this really strong negative charge, mostly in the resting of the cell. The positive charges around aren't the ones that can like interact directly with the phosphate. It's only when you start to release like calcium or something else that happens. And so something I allude to in my paper, and this is an idea that was basically built out by Gilbert Ling, who's a huge influence on my thinking about the cell biophysically in his works. That if you Google Gilbert's Ling, the first thing that will come up is probably like a Quora answer about why do scientists not take him seriously and calling him a crackpot. And right, the most interesting kind. And so what was so strange to me is I came across Gilbert Ling because he talks about how ATP and potassium and sodium and all these ions work together in the cell in a way where I'd never even heard that posed in my biochemistry. That's what I was a structural biologist now with a PhD, so I had some intuition on how these interactions happen. And so as I was reading Gilbert Ling, I was realizing like this guy isn't insane. He's actually talking about something that we really don't appreciate here. And so he actually poses in his work that when ATP binds a protein, when it forms this direct binding, so I'm talking about like when you push all of the waters out of the way and it binds to something directly, that negative charge actually can attract or like pushes the electrons in the protein it's binding farther away. And this sounds odd, but because the protein is a polymer, there is a way in which all the bonds are connected where the electrons can be pushed or pulled away from ATP as it comes close to you or pushed away. And so there's a few weird things. First of all, electrons moving in a wire and being pushed and pulled, that's how we make light. That's alternating current in our thing. So then when you have like as Gilbert Ling describes it, waves of ATP binding and unbinding. There's weird applications for understanding origin of biophotons or some illumination from this. But there's other practical chemical considerations, which is that when that happens, how strongly you are to like bind a hydrogen ion. And so in proteins, we call this kind of the isoelectric component, or the KD for the, it's basically, we're talking about how likely you are to, as a protein or a chemical group, take a proton from up in water and just add it to your chemical structure, right? And so in hydrochloric acid, chloride does not want the proton around at all, so it's released and it goes and becomes acid. But when you have something, an organic acid like acetic acid, half the time it does like to have the hydrogen on it. So it's not as strong of an acid. But when you have AT bind to something, you actually change the KDs of all of the other chemical groups of the protein. You change its attraction to hydrogen. When ATP is split into ADP because of an energetic reaction, now where you had one particle and one osmotic pressure, now you have two particles adding to osmotic pressure, the ATP and the ADP. And these will all have different KDs, which is going to affect the electrochemistry different because they'll be pulling and competing for the hydrogens differently now. And so what I've done is not made the world clearer for anyone. In fact, I just made a huge morass, right? I've just thrown so many details at you and said they all fit together, like some guy with a bunch of strings on a thumb board or, cork board or whatever. Right. And the issue is that that's where this is at, right? This doesn't become easy or like a nicer story. It becomes complicated and fractal at the edge. But these biophysical forces are there. They are manifest. And at the scale of the cell where everything is so concentrated, in a dilute test tube with infinite water, this may not make sense. But in the cell, the hydration shells are overlapping. They're all talking to each other by the pressure of these hydration shells pulling on each other and communicating this information. This starts to really have a lot of ability to manifest as interesting phenomena, right? And this might be that underappreciated aspect when trying to understand what doesn't make sense about the cell as we're looking at it. And what's left as like hanging threads in biology.
Super interesting. I can't wait to see what comes out over the years about that. Thanks for sharing. Now I'd like to zoom out for just a second and take a look at the picture as a whole and see how it might shift our understanding based on what we talked about for bioelectricity research, especially around coordination, regeneration, and morphogenesis. Do you see these topics being related at all?
I do. So I think that this is a really interesting kind of like frontier that we're poised to. We're kind of like, I think as a field, there's some interesting findings. So let's start off with bioelectricity research and morphogenesis. So my exposure to these words is basically through the work of Michael Levin. And I think that Michael Levin is becoming really ubiquitous and well known in this kind of like podcasting space because he really is just, like there's very few people who, when I listen to speak, I'm just like, wow, I'm learning a lot at every sentence. And Michael Levin is one of the most like dramatically world transforming hours you can spend to listen to him speak about biological systems. And this was really wild to me because here I was professionally studying biological systems and he was presenting stories that made no sense to me, right? One of these might be his flatworm, where if you cut a flatworm up into 200 pieces, each of those 200 pieces will grow a full flatworm. But now what he did is he made a two-headed flatworm. So where there was a head and a tail before, now he's got this two-headed flatworm and he cuts them up into 200 pieces. What do all the pieces grow? And the answer is weirdly a two-headed flatworm, something that the genetics have never encoded for. So suddenly, this weird memory is capable of overriding what genetics might consider the genetic code and what that would lead to. So Michael Levin looks at how bioelectricity is related to these memories, or bioelectricity is related to organizing biological systems and accounting for strange behavior that maybe our genetic angle can't explain in its entirety. So I became aware that these questions and the strangeness that he posed, Michael Levin always happened to be working in cells and above. So as cells get together with other cells, and this bioelectricity talks about is membrane potential between other cells and flashes of electricity signals across cells. What I happen to be looking at is from the cell level and below, right? I'm talking about organelles, how organelles and liquid phase separated or compartments in them, or membraneless organelles, have electrical properties as well that are dictating their organization at the subcellular level. So that's where I hope to add some insight into these questions. But then um, these stranger concepts like morphogenesis is this idea of like, what does the worm grow up into? How does it know going from just an embryo with genetic information and unfolding into that particular organism? How does it know to be a two-headed worm versus a one-headed worm? What is that memory that's stored when you form a seed from a plant or from a human, right? On some level, it forgets everything that the parent did and starts over from the embryo and goes through into its own world. But at the same stage, it does have some memory before what it inherited. And Michael Levin's research really complicates what that looks like for us as scientists, right? We just don't have good answers yet for this. Uh, but what we do know is that electricity matters, right? We're talking about bioelectricity. We're talking about electrochemical gradients that our cells are using and harvesting. So when we talk about like a bacteria navigating the world, we might use chemotaxis, as in it's finding the chemicals and moving towards them. So chemotax towards sugar and chemotax away from poison. We have that same version, but we also have like stranger and electrical things too. There is entire areas of agriculture, like where you're electrifying your soil, you're making electrical gradients with the plants and trying to encourage growth or discourage growth, whatever it might be. So we're now at this point where we realize that electricity and electrical fields matter. We don't know much about how they influence the systems or how they're like manifesting, and that's what I'm trying to hope to connect with my research is what this concept of electricity looks like at the molecular level, kind of given my background in protein structure. How does it look like to have electrically relevant water? And so I don't have a lot of answers for it. I just know that what the world has shown us is that this matters, and we don't really have good intuition for how these concepts apply to biology. And I'm trying to show or share some of the intuition I've developed on how I know these things manifest in these systems in a way that hopefully in the coming decades we can turn this back into understanding how things like morphogenetic fields show up. What are they, and how we can work with them.
Yeah, hopefully you two will get a chance to collaborate because it seems like you're approaching a similar idea just from different directions, but that you're very related at the same time. Because, at least from where I'm sitting, it looks like what he discusses, what you're discussing, it acts as a way to kind of upstream from genes to alter them, to change the outcome. How do you see those things playing a role? How do the concepts you've been studying affect genes?
Absolutely. So this is the cool part where in my review that we're talking about, I kind of enter in as I'm a biochemist who studied protein structure, and so I kind of show how in this field of phase separation, liquid-liquid phase separation, membraneless organization, just that thermodynamic imperative to organize in this strange way does lead to electrochemical gradients. And so suddenly now I need to know what are electrochemical gradients doing, and start to kind of like build the cell back up with this new knowledge of what electrochemical gradients are and how this matters. And so earlier I talked about osmotic balance between the nucleus and the cytoplasm as being really important, where the nuclear envelope is a semi-permeable membrane. So what is going on in the nucleus, that's the genetic expression of the cell. That's the proteins in DNA, or like that's the proteins that the cell is making to do its stuff. And so Michael Levin realizes like he has immense control over that by changing the bioelectrical state, right? He can grow a functioning eyeball on the back of a frog by changing the membrane potential of these cells, right? Not necessarily by changing the eye gene. So what matters more, the eye gene or the thing that calls it to action? So that thing that calls it to action seems to be this electrochemical potential. So what is that, and how is it communicated to the nucleus? So I start to share in my paper how osmotic pressure need to equilibrate and how electrical need to equilibrate results in electroosmotic potentials being equilibrated, and how this has to go from outside of the cell through and across the membrane into the cytoplasm and the membrane potential, and then into the nucleus. So when we're considering gene expression in the nucleus, it is going to be mediated by transcription factors. And this is a word for proteins that bind DNA and regulate the transcription, which is the turning of DNA into RNA messages. So what is turning our DNA into the messages it needs to throw out there is all about transcription factors binding, or the motors that make RNA binding and where they go. Well, where do they go? It goes where the osmotic pressure and the electrochemical potential push them, right? That's what this liquid phase separation is telling us. They're not these like motive beings necessarily with their own will. They're being pushed around by these gradients. So then as I change the pH of the nucleus, or that then I can change the binding of which transcription factors bind where. I can force them to come on and off, or just by changing the pH and the osmotic potential. So normally we are really care a lot about this because we want to grow stem cells into the cells we want them to turn into, right? For all of our treatments, we want to make sure that stem cells don't grow into cancer cells and that they grow into like the neurons or the cells you need to heal some traumatic injury or produce the products that we want. So oftentimes scientists will do this by adding proteins called transcription factors or these things in because they want to just push that binding to that right part of the DNA to turn on that program that will make it grow into like a neuron or something. But I'm saying that you can get that same consequence by just manipulating the osmotic pressure and the pH, because that's really what's controlling the on-off, right? So introducing a protein, even if it can't bind, you're not doing a lot. So I try and bridge a way by which we can go from these changes in like bioelectrical state, which is a membrane potential or pH, and understand how that pressure change manifests in where proteins are moved through these like connected liquid environments or liquid phase separated environments. And that ideally should give some intuition, some new ways to think about the cell's behavior. And there was a bit I wanted to talk about, I think, when we're talking about discussing a change in hydration state and a relation to cellular disease, how we can start to take these ideas that I've laid out of pH and osmotic pressure and them talking to each other is, and I talk about how this dictates where transcription factors and the DNA binding proteins will go that carry out a cellular program or respond to the environment. So when I was doing, so in like a cancer cell is a cell that is going to be dividing. A stem cell is going to be a cell that's dividing. A cell that is a normal, healthy part of your tissue, like a liver cell or kidney cell or neuron that's doing its job, has had its fate already set. It's become that kind of cell and it's not dividing anymore. Right? So there are certain biophysical hallmarks of a cancer cell. They're going to have a certain metabolism, a certain pH, and this is going to be inverted compared to a healthy differentiated cell. So you can imagine how the biophysical state that makes you as a stem cell and able to divide is going to be kind of similar to biophysical state that allows a cancer cell to manifest. And this is a really hydrated cell. You can think of it as a muscle cell, as like a deeply dehydrated cell, right? It's now put tons and tons of protein into it, and is a different percentage of water than a stem cell precursor is. And furthermore, when you have a cell that's dividing, you have the nucleus and where the chromosomes are, and you have the cytoplasm around it. You have to dissolve the nucleus and then divide the chromosomes to different parts and then reform the nucleus. So as I've been talking through my work, I've kind of referenced how the nucleus can be looked at as its own phase-separated liquid phase, but to become the same one as the cytoplasm, that difference that makes it the nucleus has to disappear, right? So you have to now become one phase where you were two. So usually the nucleus is a little bit more acidic than the cytoplasm. That has to disappear for this division to occur, to abolish that, right? And so things that you want to divide or don't want to divide, you can actually play with the physics needed for that to occur. A slightly different example is that in the context of amyloid diseases, coming back to where I was, when I was doing X-ray crystallography and looking for structures of these proteins, the idea is to find out where every atom is in this fiber so I can make a drug that goes and dissolves the fiber or something like that. What's strange is that in 2017, this drug study was published where they actually used the immune system antibodies to bind to these fibers so that they would be cleared by the immune system, and the body got rid of these fibers as you could image by MRI or PET scans, I forget. But the issue is that the patient's symptoms with Alzheimer's disease didn't change, right? So I was looking at this protein fiber consequence of the disease, and it was always tied to being what's wrong with the disease. And then this group got rid of them relatively successfully, and it didn't seem to change the outcome. It didn't seem to change the symptoms of the patients. So what are we really looking at with this disease? And another point that I highlight is that in the cerebrospinal fluid of people with Alzheimer's disease, it seemed as though their pH was slightly more acidic than the healthy control patients, right? So what I'm trying to point to is that what pushes a protein into a fiber is going to be a change in osmotic pressure and the pH gradient, the pH or the electroosmotic conditions available that will push it into a protein fiber. It doesn't necessarily just happen. And so then when we're looking at this disease, that should be primary. What happens first is not the protein forms a fiber and then you get the disease. You have a disease state that has a pH and osmotic pressure that is associated with it that then encourages this fiber as an outcome. So trying to get back towards like the more primary cause, what's happening first. So now you might check for acidification of the blood or cerebrospinal fluid as a way to diagnose the disease, right? As opposed to the presence or absence of this protein that may be really downstream of this more primary cause. And in this way, I pose that there is great utility in this view that I'm putting forward in trying to interrogate disease and how it goes awry from normal healthy states by looking at just these simple biophysical constraints.
Two questions came to mind while I was just listening is if you attempt to target an aspect of that to prevent disease, is that possible since all the other systems are kind of working with pH and osmotic pressure? So that's number one. And just briefly, was there ever, I guess, a hypothesis given for why when the amyloid plaques were removed, the systems persisted?
Let's start with the last question about the amyloid plaques and then remind me of the next question after. And so so strangely enough, this drug that doesn't make you symptoms better but gets rid of the protein, I think it's FDA approved, which is maybe strange considering the efficacy or the data shown for that. So that's kind of an allied thread in the field. But the reason given for why it's not is that the damage has already been done. You cleared the plaques, but the voids that they left, right? The damage, the dead neurons are not necessarily recovering. So maybe it's just too far down where that is it. And so the advocacy for why the FDA might have approved this is that if you can start this like 20 years earlier and just prevent those from showing up, maybe you can have better outcomes 20 years down the road. And this is legitimate science, like we should know this, right? So that's why these plaques may not be tied to the actual disease and why things may not get better with their absence. Can you remind me of the first question you asked?
If you wanted to treat or prevent a disease in terms of affecting the osmotic pressure or just the pH, how would you do that since everything seems to be so connected in a way?
Yeah, this is a wonderful question that I don't have the best answer on, but it gives me a couple of things to think about. The first is that why do we not do this or consider this already, right? Like you are what you eat. Why aren't we changing our diet for this season? A common reason that scientists will do or medical professionals will cite is that your blood is so strongly buffered that it's really hard to change the blood's pH. Right, so it's got so many molecules in it that will fight and increase or decrease in pH that it just is like really hard to consider like eating a more basic or acidic diet. It's like changing that. But something that we also know is that for people that do, I forget the terms now, but like entering ketosis for a diet. So you have different biochemical pathways in your body. So if you just don't eat sugar and you just rely on the metabolism of fats, you can live your life here. But you can live your life and people will choose to do this, especially for like weight loss and feeling mental clarity, certain things like that. And when you do this, your body enters a place where you are working with a different kind of energy source than the typical glucose, and you start to use a lot more ketone bodies. And a consequence of this could be ketosis, which is I don't know if it's an acidification or it is a biophysical consequence in that the makeup of your body changes. You're using different pathways for energy. And so you can live in a diet and consume a diet which can cause significant changes in the chemistry of your body, as evidenced by this. And this can be poisonous in the case of ketosis, or it can be really beneficial in the case that many people have like fantastic weight loss or experience mental clarity as a consequence of always being in this biochemical state. So we know that you can change this by diet, but we also don't really know a lot about how or why or different. I don't think that a lot of scientists actually have approached this yet to start to tease it apart. But yeah, would like a strangely acidic or basic diet predispose you to certain kinds of cancers or others, or certain kinds of neurodegenerative diseases or others? I think that this is maybe not often thought about or taken seriously, but now we're at a point where we have enough hard science and some stuff that I showed, like at the cell level and below, why we should take this seriously and consider it. And
Not to complicate matters, but have you gotten to the point in all your research, since you're zoomed in so much on the molecular level, uh, to start wondering or asking yourself, what is an ion? What is a proton really? And how does it connect to, matter, information, all that fun quantum physics stuff?
yeah, I kind of came about this journey because I came across Michael Levin by listening to podcasts during the pandemic, in particular Kurt J. Munkel. And so I was exposed to a lot of ideas there, which are asking these questions. And I was so curious about them because these are the same kinds of questions that I'm trying to understand. Like, what is charge? What is electricity? How are all these things communicating the stranger things that these scientists talk about? What is connecting them, uniting them, and how is that happening? So what is kind of interesting is that before I went on this big journey and dive, I was really struggling with the science. I actually kind of thought that uh, I wasn't sold on the importance of what I was doing. I couldn't find a question to ask in lab that reflected what I learned about Michael Levin's work with trying to cure amyloid diseases or understand the stress granule. There wasn't an immediate bridge built for that. And so I was kind of discouraged by science because I didn't know what experiments I wanted to do, right? I wanted my experiments to be uh, taking into and also working with what other people had discovered around me. And if it's so powerful that bioelectricity is a driver of biological behavior, as Michael Levin and some others show, then how does just kind of finding this like protein fiber as an outcome of this process work in that system? And it's hard for me to be motivated to do science in that. But in through going through this dive and learning about water and hydration shells and how powerful it is, this kind of like missing part of the equation, it's actually focused me around to a lot more. I don't wonder what an ion is. They're very common, happens all the time. I don't wonder about a lot of things I used to. I have a lot more knowledge than I used to. I know how ions and motions and molecules turn into electricity now. Know how they talk to each other. I know that a lot of people and physicists have an understanding of this with equations, of how equations capture a transfer of energy between one system or another. I have a very different way. I have a much more intuitive how the atoms themselves are vibrating and how that vibration changes in a hydration shell, or how that is changing during these chemical processes. And so what's strange is that I don't do the same accounting for ions I used to, right? pH is not useful to me as a measurement of the number of ions. So I don't do the accounting that maybe other people would spend their thoughts on. But what's replaced is something that's like strange and a little bit nebulous, in that all these systems are talking to each other. And it, in fact, kind of becomes so complicated that Kurt J. Mungel's channel name is Theories of Everything. I really like this other quote I've heard. It's like a theory of everything is a theory of nothing, right? It's just if you've like zoomed out that far, you're not doing anything. So this whole journey and understanding all these actually really allowed me to buy back in and realize, hey, I am asking the right question. I am doing this approach in a way that I really believe in, that connects in these ways. And so I've never been so motivated, but I also am not looking for this one size fits all, right? This is all really context dependent. Are you the cell that explodes when your membrane is punctured, or are you the cell that stays together? Right? Both exist. Both have something different about them. I have something to learn from both of them. I don't need to have a theory that like elegantly explains that. I can still work with that and make a lot of contributions, because what I'm really trying to do is make things that are useful to people. And that actually doesn't necessarily look like a theory of everything, right? If you have an astrophysicist explain why you exist there, does that cure your depression because of how stars do fusion? It's not necessarily the case. And so being really aware that like this accounting of molecules is my day job, and I like that. That's cool. I have a lot of insight from that. But this is not, I also have a lot less attachment than before, as like science is a tool to discover everything, right? Things are far stranger and more contextual than I think people realize. And so the importance of the situation is the context. And then zooming back out, like that water you're surrounded by is the context of which the molecules that are acting in it is. And so just kind of like in Michael Levin's experiment where you have the same DNA, the same molecule in the two-headed frog and the one, or the two-headed worm and the one-headed worm, right? The context is still somehow entirely different. So keeping that in mind and not just getting too lost in the detail, I think has been a really nice like outcome of this, because I know now that it's not the point to like getting lost in the details. Just that you're not being a productive member of society at a certain point if you're too lost in that detail, right?
That's an excellent answer. One thing I keep circling back to as we were talking is just thinking about all the water that's moving through your body. And I don't remember the exact amount, so maybe you can inform us of that as well. But what I'm wondering is, do you think as it's moving, filtering through the body, that in some way it adds to communication while it's going through this process?
I do, and this is somewhat nebulous, and I think cool and interesting to think about now. Some ways that I think are most applicable and like brass tacks are just useful at the end of the day is that when we're talking about signaling in the body, we care about signaling because it would kind of talk about like, does your body release steroid signals or other ways for one part of the body to talk to another, stress, inflammation, all these kinds of signals are really important. We have these molecular pathways mapped out for that. And we have how one steroid might interact with a protein that's its receptor and interact something down there. But it turns out that the thing that travels the fastest through water is pH effectively, right? Hydronium, the H plus, and hydroxide, they diffuse faster than water diffuses around itself, right? So it's actually the pH change should be the first signal that reaches you in a weird thing. It can be diluted by distance, but these electrical changes are much faster than a molecule floating from somewhere else to another. So there's immediate value in understanding like what will interface with you first, and it's often kind of going to be these electrical changes happen faster. And then if you're waiting for an actual particle or a molecule to show up, that's smell that shows up way slower than seeing something or feeling the heat from it. Right? So that is one way I'm trying to think of. So the other part that you're talking about is so in this way, water can be this like the first signal that you get in a lot of hypothetical interactions. The next one is, how is water moving through our body? And this is really strange now because of there's some things that I point to, and it's hard to make a cohesive story, but there's just some interesting facts, right? Like first of all, the people will say you're 70% water. This seems true as like a baby. Baby might be 80% water, but then an old person might be more like 40 or 50% water. So your water content actually changes throughout your life and different parts of your life, but also like cycles and hormone cycles could be even faster than that, of which these are changing. Your so a strange area of science that my research like bumps into that I'm always a little bit hesitant to mention is it's possible that the blood flow in your veins is not from the heart pumping, right? There's a legitimate theory that the heart is actually interrupting blood flow. And the only way that this theory would make any sense to anyone, or they have even listened to someone say something that's so absurd, is that you have to wonder, where does the blood flow come from originally? And what's cool about my research is that at the molecular level, it talks about how water is sucked from some places to others. And so you start to get all these like interesting turbulent flows around water or molecules exchanging from one liquid phase to another. And so can imagine that metabolism at the very small scale, it is going to be turning like glucose and six oxygens into carbon dioxide, like six carbon dioxides and six water molecules. There's osmotic potentials there that can drive or influence how water is pulled through the body just in that accounting alone that maybe we're not considering. The amount of, so if ATP is the energy source of your body, then ATP involves hydrolysis, right? Splitting an ATP into ADP and a phosphate involves the consumption of a water molecule. And also, like the two particles on the end have a different osmotic pressure as well. And so all of these aspects are pulling on water in different ways. And another strange thing about ATP and its creation is that at the ATP synthase pump, that the number of hydrogens that you have to flow through the pump to make one ATP, if you do the math for about how many of those ATPs you need a day, you get the number of hydrogens that make up a volume of water, some like thousands of gallons that are being pumped through the mitochondrial ATP as protons. And that volume of water that I calculate happens to also be about the average amount of blood you pump in a day, right? So there is some seemingly inherent tie to the amount of metabolism that you have and the amount of blood flow you have. And this all pushes to kind of an interesting vortex of water literally being split at the molecular level in the mitochondrial ATP pumps and then recombined in this weird, generator. So I start to see the body as you have all these fluids moving through and there's all these like pressures, right? And another thing that's very odd about when we consider like a hypertonic and a hypotonic, so a solution with sugar in it that has a lot more osmotic pressure than a solution with no sugar in it. The water from the pure water solution, that has more potential energy. It rushes into the concentrated sugar solution. It's not that the sugar spills out into the other way. It is the pure water that contains the potential energy that is going to be entering into the other phase, trying to run down its concentration gradient, its activity gradient, rather, into the osmotic pressure. So water is this kind of like self-modal force that's always trying to like go down the energy, roll down the energy hill, and that's what's dictating this movement at that scale. So I do start to imagine the water as a strange water vortex at these different levels, and how different organs and organ systems play with the electroosmotic pressure in the reactions that they do. Your kidneys have very particular reactions that they care, and that is also a suck to like suck out water and excrete waste in certain ways. So it's going to be different than the liver. So there's all these organs that will then impart their own particular like biochemical flavors into what the fluids are doing and how they'll interact with each other, leading to the strange water vortex of your body.
Truly fascinating. I definitely have been underestimating water. It was about 2 months ago I started re-examining it, and it's just amazing. Now, when I was reading your paper, there was like a sentence or two, and I thought, I need to know more about this. So, you had written that ions can influence how light, sound, and vibrational energy move through water. Could you talk some more about that?
Yeah, so I have this bit because uh, how I originally came across Michael Levin on Kurt J. Mungel's channel was actually because I listened to some interviews about UFOs or strange physics from this. And if you ever spend any time in the UFO world or the strange physics, there's always like a shady character at the bar who hands you a napkin with the secrets of the universe, or Tesla who has his thing. And it's always the same. It's always some version of like sound, frequency, light. Uh, and so no, and so what is sound? What is frequency? What is light? And how do we make something useful for that actually, other than just like a weird plot point to entice us into a story? And it turns out that they do actually have a really strange interaction from each other, and it's a really valid way by which to look and break down the world around us. And it's that sound is the organization of atoms jostling into each other, and we call this heat, right? And so if I heat up molecules, uh, you will see like if I take a sword or iron, put it in the fire, it's going to go from like red to yellow to white, right? in the light that it emits. And the only thing I'm doing is I'm pumping heat or sound waves into the iron, and they're exciting at a point where then they start to produce light in the red and white spectrum. And so I'm saying that sound is atoms, and then light is this other form of energy, and frequency is the mathematical tool that we use to turn one into each other. So in my work, I have this thing called the Fourier transform, and this is if you hear a song, the Fourier transform is the mathematical tool that you put it through to break it up into what each frequency is happening at that point in time. So it's a complicated sound wave, but it's composed of maybe a drum and a guitar and a vocal sound, all with different pitches and frequencies on top of each other. So the Fourier frequency is the Fourier transform is this mathematical function that allows you to understand how those are turning into each other, which are the base frequencies. So in the concept of sound, like vibration and light, you have sound, which is one version of energy, the heat stored in the iron. You have light, which is another version of energy, that thing that's being emitted via light, and it changes based upon how hot it gets, right? So frequency is what's converting. So when you have a resonant sound, it emits a resonant light frequency. So frequency is just how you convert sound frequency into light frequency. These things talk to each other all the time. So when I'm looking at fluorescent molecules in the cell with my microscopes, I actually am blasting fluorescent light into this molecule, or I'm blasting light into it. And then when this molecule absorbs that light, it undergoes a rotation or a change in its structure as it captures that momentum from the photon. And so that actually moves the molecules, right? That is heat and sound vibration. And so this will happen at particular frequencies. So some photoreceptors will accept, on average, when they accept the photon, it'll take time for them to release it. And that's what I see in the microscope. That could be maybe 3.2 nanoseconds or milliseconds or any number, right? So frequency is how you convert from resonant sound into resonant equivalent light. And you can do this energy accounting. We do this all the time. So you can kind of look at the world and bin it into what's the light energy and what's the sound energy. And frequency is how you convert these two into each other. So when I'm doing measurements as a scientist, we're almost always using this concept in our measurement. So for example, the spectrophotometer is one of the most common and useful tools in science, and it basically just means I'm measuring color. And so whatever color my solution is, I can tell by how colorful it is or what color that is, what are the light producing molecules that are resonant with that color, right? So if I have a yellow solution, there's a yellow dye in my solution. And so I'm measuring kind of how much yellow dye there is. So the spectrophotometer, I'm turning this light interaction where it absorbs light, converts that into sound and heat in my test tube as a way to get the concentration or change of what's happening. So almost every tool that the scientists use is measuring some version of sound or some version of light and converting those into each other for our intuition. And so in water, this is what infrared spectroscopy is. If you look at it, right, if you're by a heat lamp, an IR lamp or something, it feels hot because the photons that are emitted off of that IR lamp happen to be at the resonant wavelength by which your water molecules absorb it. And that means that when it's absorbing it, you're basically pumping the atoms on the water molecule to spring back and forth faster. So it's absorbing the momentum from the infrared light and converting that into heat by moving faster. And this is also why you like IR lamps. Why, if I look at you in the infrared spectrum, I'll see the body as very bright. And this is also what our instruments show. So I can look at a phase separated droplet or solution and take an infrared picture. And because the water molecules will vibrate differently in the dense liquid phase than in the bulk liquid phase, they'll have different colors. So there'll be different resonant frequencies associated with the dense phase where there's more proteins absorbed in it that will change kind of the IR light that water will accept, versus the water on the outside in that bulk phase will have different resonant frequencies for infrared light that it accepts. And I can literally measure this in lab and use this to talk about concentrations or changes in behavior. So this is how I talk about whenever you dissolve something in water, you change how the water vibrates. And in particular, we're talking about the hydration shell. The water in the hydration shell vibrates differently than the bulk water. That's why we can see it experimentally. That's why it's functional to us. And that's why these molecules will like interact with each other differently. Because what determines an interaction, the particles don't see each other first, right? They see the other particle's hydration shell before. So you run into the other person's hydration shell. And what this case says is kind of like how it changes how energy flows through it, how the water in the hydration shell has a different resonant frequency with light or a different vibrational behavior than water in the bulk. And so on some level, this is the base difference that we're looking at and trying to understand with when maneuvering through these biochemical systems. So that's its kind of like boring, like useful practicality outcome of that, of what we're looking about that.
Through reading your paper, just doing some research, and then our discussion today, I'm beginning to get this feeling that water is acting almost like a master regulator in the cell and in the body. Do you think that's too strong of a statement? How do you see water in terms of operating upstream of things or just general in terms of control?
In terms of control, so I think of water as I can definitely justify the use of the term like kind of master regulator of the cell. And ways in which I could maybe invoke that is that cancer cells tend to be more hydrated than differentiated stem cells, or a neuron or muscle cell that's less hydrated. So um, in a sense, like when an embryo or like a young human is born, they're more hydrated, and a person that dies is less hydrated. We can kind of then even look at the dehydration of their organism or the dehydration of the cell as it's differentiating as this huge determinant of its life and what it's doing. Uh, but I also think that this is kind of like one of those things where it's too big. It's a theory of everything. It's too big. It doesn't really cover the context when we look closer. So in that case, it kind of breaks down as a master regulator and how I can usefully break it down to other situations. But even then, at those like broken down atomic ways, right? Like ATP turns into ADP and phosphate, right? It's within the two. A water is consumed in that process. Water is ejected when you put them back together. There's some accounting there too. That's it's a regulator, and in the test tube, water is so abundant that we don't care about that accounting. It just doesn't is not measurable for our experiments. But when you get into the concentrated environment of a cell, like a muscle cell, I don't think it's appropriate to say that we can't care anymore about that, right? There's something that has a chemical consequence when you suddenly increase the osmotic pressure by breaking up a bunch of ATP for a muscle contraction, right? There's something going on because the hydration shells are so close that they can talk to each other and transfer information between them faster than we might normally like consider. So it's hard for me to place it as this master regulator, but I would argue that in our pedagogy and how we think of the body, it's underappreciated, often invisible, and not considered forefront. So I'm trying to put forward that this is there. And the weird thing is that you don't have a biological system without water, right? Weirdly, when you have proteins that are just a dry powder, just completely lyophilized, like enzyme, like a lysozyme, uh, it's not moving. When you look at it spectroscopically, there's no motion, right? This is the equivalent of like having that, iron rod that you heat up in the fire and changes color. You're not seeing any color come off of these proteins when they're just sitting there. But then as you add water, you get to see that they do start to move, right? The water allows them to move in ways that they couldn't when they were dry. And what's weird is that that movement shows up where like maybe one hydration shell in the lifetime or two or three, right? It's very little, but the two without each other, without the protein and the water together, each of those substances has such a different behavior, right? And so it's always the marriage of the two together that's the important part. And our contemporary view, we tend to make the water invisible, even though it is half the equation. So I'm not advocating to elevate water above 50% of what we care about when looking at the body, but I am advocating for it to consider it up to 50% of like what we're looking at in biological phenomena, because it takes both. It takes the water that's dissolved in the thing that you're dissolving in it to get the behavior we see. And one of those we typically don't consider strongly right now.
So, one thing I'd like to explore with you next time, and I appreciate you coming back in order to do this, is the stranger territory that gathers around a subject like water. Before we do that, do you think there's real scientific value in examining older or more fringe ideas even if they ultimately do not hold up, simply because they can sharpen our questions and expand how we think.
Absolutely, as a blanket answer. And I think that when talking about the strangeness or the anomalous properties of water, in my publication and in our conversation here, I talk a lot about what we can really measure and know scientifically and what is already known about that. A stranger example of an experiment people have done and we'll talk about with water strangeness is like crystallizing snowflakes in the presence of people with different emotional states, right? Does a snowflake look different when you're angry? Does it look different when you're at peace? And these things I think that the scientific mainstream doesn't hold in high regard, doesn't really approach. Uh, but in my water research, I've come across strange things that are scientifically known and highly regarded. One being, right, when people talk about a memory of water in homeopathy, this is usually discarded. However, in the crystallization of strange types of ice, there is a weird story where we normally think of ice as ice age, hexagonal ice in our ice cubes, but as you fiddle with temperature and pressure, like have a really high pressure, but and also a high temperature, you can still get ice, but it'll be organized differently. And this is one of the mysterious parts about water is that we have, uh, if you go to Wikipedia and look at ice phase transitions, we have like 20 or 10 to 20 different ices that we know of, structures that it forms. So I think that if you move your system from through temperature and pressure and go through like ice, one type of ice will always be made if you go from like through ice 4 or 5, and so you always have to go through ice 4 to get to kind of ice 5. But then if you melt that system and then you go through it again, recool again, ice 5 will actually crystallize before ice 4. And you can get rid of this by boiling the water. And so something has to go through 4 again to get to 5 as opposed to skipping it to 5. So there is a weird memory that has been documented. And this is usually considered with crystal seeds. And so you, by boiling it, you get rid of the crystal, the tiny microscopic crystal seed that's there. So we have turned this down back into like material explanations. But I think that there's a lot of opportunity to explore these like stranger people findings that people talk about. So there is a weird memory of water. There's also an interesting paper by Professor Dor Ben Amat who talks about water. The structure of water is exactly distorted as much as the hydration energy of what you dissolve into the water. And this is just actually, it's very interesting, but it basically is a perfect like physicality of what you put into the water. However, the water hydration shells are changed is a function of just how strongly energetic you put what you put in for it to hydrate. So there's this energy equivalent between what you're putting into the water and how it's distorted. And again, maybe this deserves re-examining in this case of like crystallizing ice and this strange emotional behaviors, because we can again make this much more grounded and pragmatic. When you're in a different emotional state, you have different chemical reactions happening in your body. This means that you have a different frequency. But what I mean with that, it means that the biophotons you emit will be slightly different. The temperature of your body might be slightly different. And then if water is exactly distorted exactly as much as by like what it receives, then the photons the water is receiving will again have maybe some information that's carried with them in that way too. And so we can re-examine some of these stranger findings of water, but with a much more rigorous material perspective. And I expect that this won't ruin the beauty or the strangeness of these things that people will talk about. It'll just give new meaning in how we can potentially even utilize this or take it more seriously, or find that we can't take it seriously because it doesn't hold up appropriately. And as far as re-examining older ideas, all of my work comes from building on the shoulders of giants. In particular, if Gilbert Ling had not tried to reimagine the cell as a biophysical system as opposed to a like bag of enzymes with genetic programs inside, then I wouldn't be here today. And so I really was able to inform my view through Gilbert Ling's work, which was published first in the 60s. Right? One of the creators of the MRI cites Gilbert Ling as the inspiration by which to look for contrast in the human body by looking at water, the colors of radio waves that water nuclei absorb is what an MRI machine it is, what nuclear magnetic resonance is. And because Gilbert Ling talked about how these different hydration shells form around these different types of molecules, these creators of the MRI were able to be inspired by that to make something that looked for that as contrast in the body. And these people have been largely forgotten about with how they view the body in today's teaching. And so I had to come across them. And so something that was written in 1960 as cutting edge, I found to be far more powerful and moving than any contemporary sources today. Right. So I think there is great power in revisiting history, not just for hanging threads that weren't explained. That's interesting too, but also just a really uh, understanding how they're approaching their problem and what was going on at the time. The context of society changes, but you can more reliably hunt down origins of ideas and have empathy for their creators and why they were putting those ideas forward. So it doesn't sound crazy by kind of actually tracing the chronology of these ideas too. And I think that exercise is the really important one that a lot of meaning comes from in particular.
Well, I so appreciate your time today, and I'm very excited for when we dive into topics like that and more. Thank you so much, Dr. Hughes.
Thank you very much, Dr. Smith, too. It was wonderful to uh, go through and be able to talk about and share this paper and this work I've done with you, and for you to engage so deeply and ask the questions that you've come across. I am really excited to be able to share these concepts with the world.